Yeah. Ha-ha. Now I'm just madly cackling into the camera for whoever just started. Why is she laughing? Don't tell them what we were laughing about. Okay, for people on the video, I forgot to start recording. You missed this slide. It's not a big deal. Okay, so does anyone know what integumentary means? For no points from my undying respect. I'll tell you, I had to Google it. And it's written in my notes here. Yeah?
Skin and appendages acting to protect body.
Feel strongly that you Googled that. Is that, or is it in the
It's in the PowerPoint.
You guys are real cheaters. But you know what, that's good. Okay, so yeah. That's just a fun fact. There really is nothing to do with what we're talking about. And it's the only one that's not in this black box here, so we don't really care about it. But it's just a fun fact. Integumentary. Now we know. Okay, so all we're really going to be talking about today, not all, because it's super exciting, is how we regulate pH in the body. So this is a good place to start, which is why do we even care about this? Why should we all really care about this? So this is a critical component of homeostasis. So as you know, like inside your cells, you're wanting a whole bunch of metabolic pathways to work properly, and pH is a very important part of everything working properly. So does anyone know the ballpark of what your pH is in your stomach? So it's like hella acidic, basically. I'm going to give you the pH in a later slide. But the idea behind like a really acidic stomach is you can eat, I was going to say almost anything, and your stomach acid will break it down because acids break things down. So you can imagine if for some reason your cells were to get acidic, and not even as acidic as your stomach, but just more acidic than usual, you start having changes to the shape and function of enzymes and proteins. They basically start getting broken down, and you're really hooped. So it's important to keep pH inside a normal physiological range. And then of course, optimal metabolic function, so the enzyme-driven chemical reactions, which are going to depend on the structural integrity of the enzymes and proteins, is also going to get affected by changes in pH. So it is really important, even though it's boring. I'll stop. That'll be my last comment. This is boring. I'm really salting my lecture here. Okay, so acids are any substance that dissociate in solution to release hydrogen ions. So you've got lactic acid, which is the one that we're going to be talking about a ton today, or one of them anyway, that we're going to be talking about a lot today. So you plunk lactic acid into solution, and it's going to dissociate into a lactate ion and a hydrogen ion. So, and a hydrogen ion, actually, I'll save it for the next slide. And then a base is any substance, so basically an acid is a hydrogen ion donor, or proton donor. And then a base is any substance capable of like accepting a hydrogen ion, or combining with a hydrogen ion. So bicarbonate, for instance, which is this HCO3 with a negative on there, going back to chemistry, that can combine with a hydrogen ion to create carbonic acid. And so by accepting these hydrogen ions, it decreases the pH of a solution, because hydrogen ions, again, are only going to affect the acidity of a solution. I say again, okay, we're covering this on the next slide, when they're just free-floating around in amongst themselves. I was about to make an analogy of like a single person at a bar. It's bad. We'll carry on. Okay, so base can accept hydrogen ions, acids donate hydrogen ions. Acids donate hydrogen ions, and then bases can then accept. Yeah. I'm beleaguering a point. Does anyone have any questions? We'll move on. Okay, so I should have done this first. So pH refers to the hydrogen concentration of a liquid solution. So the concentration of protons. So the pH of the solution is defined as the negative log of the concentration of protons. So basically, like, it's a logarithmic scale. So one unit change in pH corresponds to a 10-fold change in protons. So here's neutral pH, where you have an equal amount of hydrogen ions to hydroxy ions. That's right? Yeah. You're not on your file. Okay. And obviously, if these two things combine, they just create water. H2O. Okay? So then the more protons you get, the more acidic you get, and the lower your pH gets. Have you guys learned all of this hydrogen pH? Yeah, okay, good. I was going to say, if you're confused between the relationship between pH and hydrogen ions, now is the time to like get it into your head that more hydrogen equals lower pH and more acidic, because we're really going to skim over that like a whole bunch of times in the lecture, and if that's confusing, you're really hooked. Okay, so lots of hydrogens, an increase in pH, an increase in hydrogen ions causes a decrease in pH, acidity. So the pH of bodily fluids range. So your hydrochloric acid in your stomach is 1. Again, like highly acidic, way at the top of the chart here. This actually, I just realized, is a terrible chart because it doesn't actually have pH on it. But you get it. I think it explains. I put this in. Dennis didn't have it in, so it's my fault for putting a stupid figure. But at least it shows the concentrations of hydrogen, and here's a pH that's really low. Here's a really high pH. And then your arterial venous blood is around 7.4. Et voila. Neutral pH is 7, so that scale, which didn't have a scale, you can draw it on if you want, goes from 1 to, I want to say 14. Yeah, that sounds right, thank you. So I put that scale up, like kind of foolishly, realizing there's not a scale in there now, to show that what we're oscillating around in terms of like normal pH in the blood is not 7. So that's a neutral pH, but our sort of normal, healthy, everything's functioning in that copacetic way is actually 7.4 plus or minus 0.5. So in between 7.35 and 7.45. This is our happy pH in the human body. And this is about 40 nanomoles per liter of hydrogen ions concentration. You guys also know this means concentration, right? Yeah. Dope, dope, dope. Okay, so then obviously you become more acidic, pH is going down, hydrogen ions are going up. This is called acidosis. And then if you head the other direction with less hydrogen ions, I might screw this up at some point. It's a lot to say. Less hydrogen ions, pH going up, so you guys get it if I say it. Just look confused at me. But that's alkalosis. So less hydrogen ions, higher pH, and you've got a more basic solution up at this end. Why do we call it alkalosis instead of like basicosis? I don't know. I'm going to have jokes like that all day because this is, again, quite a dry lecture. Okay, so acidosis, pH less than 7.4. This is the accumulation of acids, loss of bases. Alkalosis, greater than 7.4, accumulation of bases. Once again, acidosis is different than like an acidic solution. Something that's acidic is less than 7, that's neutral. So remember, those two things are different. The human body doesn't sit at a neutral pH. It sits at a pH of 7.4-ish, plus or minus 0.5. Cool? Okay, we'll say this one more time here, probably say it a bunch more times. pH and hydrogen ions are inversely related. As hydrogen ions increase, pH decreases. And arterial blood at rest in healthy subjects is slightly alkalotic. I googled that to make sure that was a real word, and it is alkalotic at 7.4. Anyone have any questions? Okay. I also had a really good sleep last night, so that's why where all this energy is coming from. I'm really wasting it on acid base now. Okay, so during exercise, the major threat to the pH of body fluids is acids that are formed during aerobic and anaerobic metabolism. So during aerobic metabolism, you get the production of CO2. We didn't just go through this, but this was on your midterm, we all get it. You're creating CO2 during aerobic metabolism. And that will react with water, which is obviously everywhere, to form carbonic acid. You see? And this through this carbonic anhydrase enzyme. So this enzyme will link these two together. They won't link together unless this enzyme is present. But when the enzyme is present, it'll create and when there's a concentration gradient, obviously, it'll create carbonic acid. And once you create carbonic acid, that will dissociate into proton and bicarbonate. Carbonic acid, proton, bicarbonate. We're going to be talking about bicarbonate a lot today. So it's a volatile acid because it just breaks apart like crazy. The CO2. Okay, organic acids. So these are organic compounds containing carbon, obviously, organic, not like at the store. My dad was a chemist and we always used to complain about how people said said things were organic, but that really actually means carbon containing. They really co-opted that word though, so anyway. Organic compounds containing carbon with acidic properties. So the organic acid that we're going to talk about today is lactic acid. I feel like we've really covered this to death. If you don't know about lactic acid, actually, your final's not cumulative, is it? You can forget all? No, you can't forget about lactic acid because it's on this exam now. Okay, so this is the thing about this class is everything sort of accumulates, so there's going to be crossover, sorry guys. Organic acids are from the metabolism of muscle glycogen and blood glucose. Lactic acid does, sorry, and that then dissociates, boop, to release, again, hydrogen ions, so acidic. So you're doing your metabolism, whether you're doing aerobic or anaerobic metabolism, you're producing acids. And these acids are going to end up dissociating and creating protons. All those protons are lowering your pH and creating an environment which, if you don't deal with it, is going to cause your proteins to start changing shape and not working, your enzymes aren't working properly. Good with this? I don't know if I have extra time to just be sitting there vamping like this, but okay. And so we call this lactic acidosis because you're having an acidosis. You're moving towards a more acid environment, a more acid pH, especially relative to our resting point, 7.4. And this is because of lactic acid. Lactic acid acidosis. It's hard to say. Okay. So, we said what's going to be a problem. We're doing our exercise. I just had to run up the stairs. I forgot my pointer. I've got lots of hydrogen ions floating around my body. My blood's becoming more acidic. What are we going to do to regulate pH? So this is maintained by chemical and physiological buffers, which resist pH changes by accepting hydrogen ions from a solution when the protons increase. Or if for some reason you have the opposite effect, they can release hydrogen ions into solution in order to bring your pH back down. So we're going to talk about 3 different kinds of buffers. We're going to really focus on the chemical buffers through probably most of the talk today. Lecture. And then we'll touch a little bit on the physiological buffers, and at the very end, we'll talk about how they all work together. Okay. Chemical buffers. So we have intracellular buffers and extracellular buffers. So intracellular, obviously these are the buffers that are hanging out inside your cells, so trying to target the acidity as soon as it happens, like in your muscle cells, for instance. And then you've got your extracellular buffers, so the buffers that are hanging out in the blood. Bicarbonate, I mentioned we're going to talk a lot about this today. Bicarbonate's in both. Hemoglobin is obviously just an extracellular buffer. Don't say that it's intracellular on the exam, because I'll laugh at you. Okay, so these are chemical buffers are the rapid first line of defense in acid-base regulation. So this can happen really, really quickly, and as soon as your blood or your intracellular pH becomes more acidic, they can actually react through a chemical reaction in order to rectify the situation. So buffers consist of a weak acid and the salt of that acid. So you've got, this is the example down here, where you've got your salt, sodium chloride, and your acid, hydrochloric acid. So these can convert back and forth between each other to either accept or donate hydrogen ions. And we'll show that. Oh yeah.
Bicarbonate is both intracellular and extracellular?
You got it. Yeah. Now let's talk about bicarbonate and lactic acid. Okay, so you get lactic acidosis. So let's say you're exercising. A real-time shot of my bicep up there on the screen. So lactic acid. This is going to go into, as soon as it's produced, go into solution, creating lactate, which has a negative charge on it, and a hydrogen ion. Et voila. Now, this will combine, I tried to make this plus sign bigger, but this was a picture, so sorry. So maybe circle this plus sign, or really emphasize it, because lactate will combine with, the hydrogen ion and the lactate will combine with sodium bicarbonate. So this is going to dissociate into a sodium ion, pretend I'm drawing it over here, and a bicarbonate ion, HCO3 minus. And then you get sodium lactate and carbonic acid. Shall I draw that on the board, or you guys get it? You get it? I got a nod. You get it. This isn't chemistry class after all. Okay, so you got sodium lactate and carbonic acid. So here you've got your salts, the sodium lactate. Here you've got your weak acid, the carbonic acid. And then carbonic anhydrase is going to, again, catalyze the conversion of carbonic acid, just like it did, I showed you on that previous slide, going this way. It can also convert it going this way. And when you break down carbonic acid, as we said before, that results in water, which is super. It's just going to hang out in the blood, and CO2. And CO2, as you can see on the slide here, gets blown off at the lungs. So you can get rid of it. And we've got rid of our protons. So sneaky. Any questions? If anyone doesn't get it on this slide, you're going to be, we're probably going to say it like a few more times, but this is important. This is basically what this whole lecture is about, this slide. Let's do it again. Okay, so the bicarbonate buffering system represents 65% of the blood's buffering capacity. So 65% is obviously, for those math people out there, like almost 2/3 of the blood's buffering capacity. So the bicarbonate buffering system is a very important buffering system. All right, so you have acidosis, which again, increase in hydrogen ions or decrease in pH, same thing. I'm the most accent-prone person in the world today. I don't know what's wrong with me. Okay. Increase in hydrogen ions. This is going to get detected by the chemoreceptors. We're going to go into this in more detail in a little bit here. This is going to increase ventilation, alveolar ventilation, which is then going to decrease carbon dioxide. So again, you're blowing off CO2. This lecture is really repetitive now that I think about it, because I'm thinking we've talked about this again in coming slides. And remember, going back to those models and really just like concentration gradients. So we're decreasing CO2 over on this side of the equation. So that's going to shift the whole equation to the right. We're decreasing CO2, so then we're going to have more conversion of H2CO3 into our carbonic acid into CO2 water. And again, we've increased ventilation, so that's all getting blown off. So as we're getting more of conversion of this over here, then we get more conversion of this into the carbonic acid, and thus you have less protons hanging around, and thus your acidity goes down. So your response to acidosis is an increase in ventilation, which really works through this bicarbonate buffering system. Does that make sense? So the ventilatory response is really using the bicarbonate buffering system to get rid of CO2 and to increase pH. Almost said lower pH. We all got that? Okay, and we have our bicarbonate ion there, that's the hydrogen acceptor. Thus an acid. I mean a base. Close one. What about the other way around? What about the response to alkalosis? So again, we got alkalosis, this is a decrease in hydrogen ions. We're going to decrease ventilation. We decrease ventilation, what's that going to do to CO2? It's going to increase CO2. So we increase CO2, the direction of this, what's the word for this equation, is going to go to the left now. So we're going to get more carbonic acid and more hydrogen ions. Boop, boop. And now we've rectified the alkalosis because we got more hydrogen ions, but we're becoming more acidic. I told you guys this lecture was going to be fun. Are we all having a good time? Does anyone have any questions about this? Okay. And now we have our acid that's donating hydrogen ions. Cool. Okay. I should have thrown in some kind of like highlights. Did you guys see the Leafs crushed it last night? Did the Habs win last night? No. Oh, that's sad. Okay. It's not really too bad. Leafs are so good this year. Okay. So chemical buffers continued. Hemoglobin. So this one represents 30% of the blood's buffering capacity. So venous blood in particular is going to buffer the blood, the pH of the blood, because as hemoglobin releases oxygen in the tissues, it increases the ability to bind hydrogen. So it has an increased affinity for hydrogen ions. So the venous blood is a really good buffer. in particular. So again, from the dissociation of the relatively weak carbonic acid, which we know is going to, again, dissociate in solution, as produced in the red blood cell from carbon dioxide and water. So again, the red blood cells where we've got the production of carbon dioxide, boop, 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 through the bicarbonate system to carbonic acid, which is going to go into the blood, dissociate back the other way. So then you get, not back the other way, in the same way, into hydrogen ions. So the venous blood can buffer those protons through hemoglobin. So the protons generated from the dissociation of carbonic acid combines readily with deoxygenated hemoglobin. Again, deoxygenated hemoglobin is the key here. This is why we're talking about venous blood. It's a deoxygenated hemoglobin that has an increased affinity for hydrogen ions. So you have a hydrogen ion plus a hemoglobin, deoxygenated hemoglobin, to create, I don't know how you say that, HHV, hydrogen hemoglobin. Any questions about how? Okay, and then finally plasma. So we have about 5% left in our math between our 65%, 30%, and 5%. So the last 5% is made up of plasma and intracellular proteins. So plasma obviously circulating in the blood, intracellular inside the cell. So these are albumin and phosphate. So phosphate again, so we have a negative ion here, hydrogen phosphate, which can obviously, I think, accept hydrogen ions. And then you get this H2PO4 negative, which can combine with a hydroxyl ion. Oh, I said hydroxyl back in the first line, crushed it. And then you have this dissociation into water. So again, we've buffered these protons and made the intracellular and plasma environments less acidic. All right, I swear that's like the most dull part of the lecture. What about physiologic buffers? Because then we're going to get into everything combines together. So are there any questions about the chemical buffers? Okay. So the pulmonary and renal systems present the second line of defense in acid-base regulation. So their buffering is going to occur only when a change in pH has already occurred. So our buffers, our chemical buffers, are sitting around, they're just doing their dissociation thing, they're following concentration gradients, it's all concentration gradients. As the chemical buffers are doing their thing, they're going to work in concert with, or if they can't control the pH on their own, you're going to activate these physiologic buffers. Okay, I picked up my coffee and didn't drink it. I'll pause while you read this. Okay, so I mentioned this already. When you have increased concentrations of hydrogen ions, that directly stimulates the respiratory center to immediately increase alveolar ventilation. Again, we're going to go into more detail about this in a future slide. But we have this increase in ventilation that happens with the increase in hydrogen ions. That's a rapid adjustment which reduces alveolar PCO2 because, again, we're increasing ventilation, you're breathing more, you're blowing off CO2 from the blood. This obviously accelerates the recombination of hydrogen ions with bicarbonate because of the equation that I didn't put back up here, but you get it. We're driving the equation back, lowering free hydrogen ions in the plasma. So that's our ventilatory buffer, physiologic buffer number 1. Physiologic buffer number 2 is the renal buffer. So kidneys, which took me like literal years of undergrad to remember that renal and kidneys meant the same thing. Don't judge me. I find the kidneys also really boring. That's like the worst part of physiology. Does anyone else hate renal physiology? Is that just me? It's so boring. Someone nodded at me. Okay. So the renal buffers are excretion of hydrogen ions by the kidneys. So this is relatively slow, but it provides an important longer-term defense to maintain the body's buffer reserve. So obviously it's important to be able to keep your alkaline reserve high so that you're prepared to buffer these hydrogen ions over here. So renal tubules within the kidneys are going to regulate acidity through complex chemical interactions that secrete ammonia and protons into the urine and then reabsorb alkali chloride and bicarbonate. So again, we're keeping these reserves high. So if anything is prolonged, again, this is a relatively slow process, but we're keeping our bicarbonate high and just getting those protons out of there. So this is something I found on the internet, and I said internet, funny, that I thought actually was super intuitive. So I thought I would throw it up here, because this really integrates all of the chemical and physiologic buffers together. So you have an addition of protons, and these protons immediately will go into the bicarbonate buffer system. You've got your bicarbonate ions hanging out here. These bicarbonate ions, as we just said, are coming from largely, or at least in the long term, the bicarbonate reserve, which, as we just said, is maintained by the kidneys over the long term. So the kidneys, again, are going to be able to secrete those protons and reabsorbing the bicarbonate ions to help replenish this bicarbonate reserve. Meanwhile, over in the lungs, if we have sort of like an acute acidosis going on, so that's a renal response to acidosis. If we have acute acidosis going on, we know that these protons are going to stimulate the chemoreceptors, which are going to increase ventilation. This is going to blow off CO2, so we again have decreased the amount of CO2, which then drives more carbonic acid into CO2, which then drives more of the bicarbonate and hydrogen ions into carbonic acid, thereby decreasing the concentrations of hydrogen ions as well. This is sort of an added slide. I guess we've been repeating stuff a lot, but I liked it. I thought it was good. Okay, and I'm teaching. Okay, any questions up to there? Okay, so now into the Henderson-Hasselbalch approach to acid-base balance. So the Henderson-Hasselbalch equation uses pH as an overall index of acid-base balance. So this is not just like a exercise phys thing, this is like acids and chemistry. Have you guys learned about Henderson-Hasselbalch in like chem class and stuff like that? Yeah, lots and lots, okay. And I wish we really didn't have to teach this, because man, I just had a rough time going back through all this stuff. Sorry, I won't complain, you guys are the ones that have to write an exam on this. Okay, so this uses the partial pressure of CO2 as an independent measure of the respiratory component of acid-base balance, and bicarbonate concentrations as an index of the metabolic component of the acid-base balance. So we're going to be talking a lot about respiratory adaptations, metabolic adaptations. When we're talking about respiratory, we're really talking about the PCO2, partial pressure of carbon dioxide, and we're talking about the metabolic component, the concentrations of bicarbonate. So this equation describes how PCO2, bicarbonate concentrations, the negative logarithmic of the apparent dissociation constant for carbonic acid, which is this pKa, it's a constant. I loved constants when I was in chemistry class. Oh my gosh, you just flow right through a constant. And then the plasma solubility of carbon dioxide, which is S, another constant, to determine arterial blood pH at a body temperature of 37 degrees Celsius. I'm not going to say that equation out loud, I'll just let it wash over you. Okay, so there it is back at the top here. So let's insert our constants here. So we have our constant for pKa, our constant for S. So in healthy subjects at rest, our bicarbonate concentration is equal to about 26. I want to say this is milliequivalents per liter. Does that sound right to anybody? Yeah, nice. I was just testing you, I knew that. And our PCO2 is roughly 40 millimeters mercury, that I actually did know. So if we plug in all of those values here, we get a pH of 7.4. Good times. This is the most obnoxious lecture I've ever given. I feel like it's very obnoxious. Okay. All right, so the Henderson-Hasselbalch equation is going to identify 4 primary acid-base disturbances, because I guess it gets a little interesting now. Again, we're talking about respiratory component of the acid-base balance and the metabolic component of the acid-base balance. Remember, we can either go acidic into acidosis, or we can go basic into alkalosis. So 4, which are respiratory acidosis and respiratory alkalosis. And I don't see anyone freaking out. I don't know if it's because you're bored, but remember, you don't have to freak out. These are just the opposite of each other. So it's just the exact same equations going one way or the other. So respiratory acidosis, remember we've got too many protons, so we got to get rid of them. And this comes about because we have alveolar ventilation which is below the metabolic requirements. Because the whole purpose of alveolar ventilation with respect to acid-base balance is to blow off that CO2. So we're not blowing off enough CO2. And remember from the equation, that means that we have too much CO2, so it's going to drive the reaction that way, or at least not let the reaction come away from the protons through carbonic acid into the CO2 and water. Yeah, so we're going to have PCO2 that's too high, again because of that bicarbonate reaction, proton levels that are too high, and pH that's too low, and you would get this in like respiratory failure, for instance. What about the other way around? So this is ventilation above the metabolic requirements. So for whatever reason, for instance, if you're pregnant, and Dr. Dennis Jensen did some really interesting research in this. I was like citing Dennis when I was in my old lab before I even knew Dennis. How fun is that? Okay, so you have this high ventilation in pregnancy, which doesn't really suit the metabolic requirements because pregnancy actually potentiates this ventilation. Progesterone. Fun facts. That's not in your exam, but it's a fun fact. So obviously, if your ventilation is too high, that means you're going to be blowing off too much CO2. So you're going to be, again, driving the reaction in that direction, away from the protons. So you're going to be reducing your proton concentrations and thus increasing your pH. Am I going over this in too much detail? It's okay. Nice. Thanks, guys. Okay, so then let's do the other two, metabolic acidosis and alkalosis. So metabolic acidosis, this is kidney excretion of bicarbonate that's too high. So you're secreting, too much bicarbonate, so you don't have those bicarbonate reserves. Remember, the kidneys are really important in keeping those bicarbonate reserves level. So you have less bicarbonate, obviously, and that obviously adversely affects your blood buffering capacity. So once again, your protons are going to get too high, and your pH is going to get real low, and that happens in kidney failure. So my kidney failure is a real bummer. And then metabolic alkalosis, so a decrease in the kidney excretion of bicarbonate, so it's not doing its job well enough. You've got increased bicarbonate concentrations in the body, you've got an increased blood buffering capacity, so you've got eating up all of these protons, increasing your pH, and this happens in excessive vomiting and diarrhea. Good times. Okay. Is everyone comfortable with the terms metabolic acidosis, alkalosis? Because we're just going to start throwing them around willy-nilly now. Okay. Sorry, I meant to change the spacing on this slide and obviously did not. So, compensation. Compensatory responses involve the system opposite to the one that caused the primary acid-base balance. So if you're metabolic acidosis, if you have metabolic acidosis, compensation will occur through the respiratory system. Again, sorry this slide looks crappy. I meant to come back to it, I did not. Or metabolic compensation for respiratory alkalosis. Or the other two that are possible. And the whole idea here is that compensation is going to move the pH towards the original normal value, which is what in a human being? Nice. 7.4 was the answer if no one heard that or remembered that. Okay, so regulation of acid-base balance by the lungs. So here's our fancy dancy equation up at the top. So the respiratory system, as we described in the previous slide, it provides the body with a rapid means of regulating blood pH by controlling PCO2. And this is on the order of like seconds, minutes, or hours. Remember, if you've got something that's compensating within seconds, it's not going to be the kidneys. They're going to take longer to do their thing. But the respiratory system can go really quickly. You know what I mean. Compensate really quickly. Okay. We'll say it all over again. You've got an increase in protons. It's metabolic. So that means we're looking at metabolic acidosis. This is going to activate the central and peripheral chemoreceptors. That is going to stimulate the respiratory control center. So these chemoreceptors have afferents that go up into the medulla, where you have these respiratory control centers. which is then going to stimulate alveolar ventilation, so activate the chemoreceptors, activate the respiratory control center, increase alveolar ventilation. We increase alveolar ventilation, and what happens to CO2? It goes? Yeah. Down. Perfect. CO2 goes down. We're blowing off all that CO2. I'll have to say it again. You guys get it right. You want me to say it again? So we drive the reaction to the right. This way. Oh, sorry. We drive the direction. Oh my god. Ignore me. We drive the reaction to the left. I'm an idiot. Oh wait. I think there's even arrows on this. No, there's not. I'm thinking of another slide. We're going to say this all over again. This is a real challenge for this lecture. Okay. So again, we decrease our PCO2, which again is going to shift everything to the left, thus decreasing our hydrogen ions. And thus we've gone back and we've fixed this problem. So the respiratory compensation for metabolic acidosis. How about I just click the buttons for the other side? Yeah, go.
Why is there an arrow from H2CO3 to the hydrogen ion?
Here you mean? Or here?
Yeah.
Well, this is just saying we're going back. It's a negative feedback loop. So pretend that we've changed this arrow to down. So like this was the stimulus for the compensatory action, like the respiratory compensation. And because we've decreased the hydrogen ions, this has become compensated for. So that this plus arrow is no longer here. Put a little like cross out. Because the problem was we had lots of hydrogen ions, but now we've got rid of those hydrogen ions because we blew off the CO2. So we had a shift in this, the whole reaction shifts to the left. So we have less CO2, so we're going to have more carbonic acid moving into CO2 and water, which means we're going to have more bicarbonate and hydrogen ions moving into carbonic acid, which means we're going to have less hydrogen ions. So we fixed this problem. Good? Oh, I was going to skip over it. Thank you. Do you guys want me to go back through this, or should I just click? You don't want to hear my lovely voice. It's just the opposite. I can say it if anyone really wants me to. You all look like you're begging me to move on. Okay. If you have a problem with this, come visit me. Okay. But we have to do it again with the kidneys now. Okay. So regulation of acid-base balance by the kidneys. We've got the same reaction up top. The kidneys contribute to the long-term. I probably could have given this whole lecture in like 5 minutes. All right. The kidneys contribute to the long-term regulation. So now we're talking hours or days, regulation of protons. Do you guys want to just like read this for a second and then we'll just like move on? You can nod when you're through it, or does this all make sense? So we have, okay, sorry. I'll say the first one and then we'll move on. So you've got respiratory acidosis. Why does respiratory acidosis happen? It's because for some reason, whatever the reason, if it's some kind of pathology, you have ventilation which is too low based on the current metabolic demand. So when your ventilation is too low and there's a mismatch there, you've got an accumulation of carbon dioxide. So an increase in PCO2. So we're increasing this side of the equation, so everything goes that way. So now we've got increased carbonic acid. When we have increased carbonic acid, that shifts this reaction towards this side, and we've got lots of protons hanging around. So we are acidic in respiratory acidosis. So that's going to stimulate a decreased renal excretion of bicarbonate. If you decrease your excretion of bicarbonate, that means you're going to have more bicarbonate in the blood. So now we're increasing this side of things. So that's going to increase the blood's buffering capacity. So remember, we had all these extra hydrogen ions hanging out because of this increase in CO2. So we had hydrogen that was just like bopping around the blood, being like, hey, I'm making things more acidic. So the kidneys are like, oh no you don't, we're going to decrease our excretion of bicarbonate. Now we have more bicarbonate, the bicarbonate grabs the hydrogen ions, and we have less hydrogen ions going around because that all moved over into carbonic acid, and we have increased our pH. And vice versa. I know that's not how you say that. Does anyone have any questions about this slide? Okay. Steel yourselves for a crazy diagram. I saw this and was just like, man, why?
You just know.
Yeah. Huh? Should we just nod? Yeah. Just
No,
nod.
I'm saying you just know it's one of his.
Yeah, it really is. Like, he loves it. And it's a nomogram. I texted him about one of his slides. I was like, what the hell is this? He loves like fancy diagrams. But actually, this one's okay. So we just have to be calm. And he gave us a nice little legend over here to work our way through it. So here, our red dot is our normal, healthy levels of everything. So normal, healthy, resting human, all of you guys right now, because you're not stressed out about this diagram at all. So we're sitting at a pH of 7.4 as per usual. Bicarbonate concentrations about 26 milliequivalents per liter. Yeah. Oh yeah, and a pressure CO2 of 40 millimeters of mercury. Okay, so this is the way you do this. ABD, so just like connect the dots. So you're sitting at A, and then you have respiratory acidosis. So you see the respiratory acidosis. Boop, you're up to B. Oh, I'm getting real cross-eyed staring at this diagram for some reason today. Okay, so respiratory acidosis. Now we're at B, so our pH has come down. Our concentration of bicarbonate has gone up. And so what we end up having is a renal compensation, which is going to, what am I saying here? Yeah, which is then going to bring our pH back. So remember okay, let's go. We have respiratory acidosis, so what do we have a lot of now? Hydrogen ions, yeah, thank you, yeah. Lots of hydrogen ions. So what do we need the kidneys to do? Make lots of what? Bicarbonate. So, I just replaced these burgers. I think I need a new pointer. So what do the kidneys do? They make lots of bicarbonate. Boop, boop. So now we're sitting up here and we have fully compensated for the respiratory acidosis. So fun. Does anyone want me to go through these one by one? Does anyone want me to not go through these one by one? I got a head shake. Yes, okay. If anyone's stressed out by this figure, now is your last time to make me stop and go through one or two of the other ones. Okay. Just scratch your nose.
I actually just have one question.
Please.
Why doesn't it go back down to the orange dot?
Well, it will, because then it's gonna, those two things are gonna combine, and it's gonna bring it back down. But this is showing what the compensation is.
Oh.
Because as the bicarbonate is combining with the hydrogen ions, then your bicarbonate is gonna get eaten back up again. So then it's not gonna be bicarbonate anymore, it's gonna be carbonic acid. Great question. Okay, I'm gonna move on. I always fly through these lectures. I don't know if you guys are talking about dentist lectures, but okay. So acid base regulation during exercise. Okay, so yeah, we haven't even really hit exercise. We've been talking about CO2, we've been talking about lactic acid. So what does everything look like during exercise? So this slide I thought I would put up. I know you guys were just covering this with Dennis, I think. I'm pretty sure you guys got to the ventilatory threshold with Dennis, right? No? Okay, it's a good thing I put this in here. So the ventilatory threshold, which you can see here at this dotted line, this is what happens when we throw this lecture in the middle of the respiratory section. So ventilatory threshold is the point where pulmonary ventilation increases disproportionately relative to the increases in oxygen consumption. So remember we talked about this with the RER and the respiratory quotient, that in general ventilation is going to be coupled to oxygen until you end up having lactic acid production, because lactic acid production, because of everything we talked about today, is going to increase, I forgot to make this too little, sorry. It's going to increase CO2, and the CO2 is going to stimulate the chemoreceptors and increase ventilation. So we're coming full circle back to our respiratory exchange ratio stuff. So the ventilatory threshold, again, it's where it increases disproportionately. So disproportionately means there's an excessive ventilation based on the amount of oxygen consumption. And this excess ventilation is directly because of the CO2 that's being produced as the body buffers lactic acid. Okay, so here we got work rate. So we're working up to percent VO2 max. So we're obviously increasing in intensity here. Here's your muscle pH. Here's your arterial blood pH. These look pretty similar. although muscle pH starts dropping right away. So as you get to around 50% of your max work rate, this is your ventilatory threshold here, because your pH is dropping really suddenly, and your pH is dropping really suddenly because of all the lactic acid production and all of the protons that are getting produced. And so, again, that's stimulating the chemoreceptors and causing the increase in ventilation. Are we confused or just bored? Bored? I've said this like so many times, right? But I shall move on unless anyone's confused here. So during heavy intensity exercise, so exercise above the ventilatory threshold, there's a progressive decrease in both muscle and blood pH, reflecting yada yada yada, blah blah blah. The increased production of CO2 from aerobic metabolism, the increased production of lactic acid from anaerobic metabolism. So again, this is happening from both aerobic and anaerobic because of the CO2 and the lactic acid. And then the amount of lactic acid and CO2 produced are obviously going to depend on exercise intensity, muscle mass, and exercise duration. Because as you know, the lactic acid is really going to get formed at those higher exercise intensities as you use more anaerobic metabolism. I got one nod, I'm moving on. Okay, so basically what does this all look like during exercise? So kind of same figure, we've got work rate across the bottom here. Here's our pH that we showed before. And here's our lactic acid getting produced. And like we could probably have had like 20 more figures in here showing the lactic acid getting produced, showing the protons getting produced, showing the bicarbonate that's getting eaten up by those, and by eaten up I mean it's combining with those hydrogen ions to buffer the hydrogen ions, which are creating an acidic environment. So the bicarbonates